right? The thing was, the mechanism that limits the number of cellular divisions is something called a telomere, which is a repetitive sequence, a genetic sequence at the end of chromosomes that shortens slightly every time a cell divides, and there's a point at which it's grown critically short and the cell refuses to divide anymore. It's called a Hayflick limit. And the idea that that Hayflick limit might be a feature rather than a bug because it prevents tumors made a lot of sense. Every mole you have turns out to be a would-be tumor that got arrested at the Hayflick limit, something that I called a prototumor, a term that's never caught on, but it turns out to be accurate. Okay, so elegant explanation. We grow feeble and inefficient because otherwise we'd be overrun by tumors, and selection has delicately balanced these things to extend our lives, navigating between these two hazards. But mice have ultra-long telomeres and very short lives. They have telomeres way longer than humans, and yet they don't live very long, so
that's
false.
The ugly fact that slays a beautiful hypothesis. Feynman, I think, said that, did he?
Oh, I don't remember him saying that, but the ugly fact. But the thing is, it's not even a scientifically legitimate thing to do. It's just a hunch. All of the questions that I knew existed in this landscape that were answered by my hypothesis told me, you're missing something. There's something you don't know. And so I stuck with it even though my hypothesis had been falsified by this one observation. And at first I thought maybe this is one of these cases where one person has measured the telomeres of mice, their experiment was contaminated, and everybody else has just been repeating the same thing ever since. Nope.
Wishful thinking.
Yeah, wishful thinking. It had been tested 12 different times by a bunch of different labs, and they all came up with the same thing. Mice have ultra-long telomeres. Like, what the hell is going on? Well, it turned out, through a series of stumbles and blind alleys, that I found the answer. The answer was mice do not have long telomeres. Laboratory mice have long telomeres. So I hypothesized that, and Carol Greider, who has now gotten a Nobel Prize for her discovery, her co-discovery of the enzyme telomerase, which adds length to telomeres, she and her then graduate student Mike Heeman tested my hypothesis, and they showed, eureka, wild mice don't have long telomeres. So that told me there was something going on in the colonies of the mouse breeders that elongates telomeres radically. And I have the only hypothesis that remains standing for what that is. That one hypothesis that could in principle explain this is that those colonies are interfered with by an economic phenomenon. Mice breed slower the older they are. So mouse breeders, in an effort to be economically efficient, only breed young animals. They throw them out after 8 months. That's not long enough for these animals to get tumors, so the selective force to keep the tumor prevention system running is eliminated in the colonies, which unbalances the trade-off. So they evolve in the direction of long telomeres because it keeps them functionally young for that entire 8-month period, and the fact that they all die of cancer doesn't matter. It doesn't have a selective impact because they've been eliminated from the colony by the time it happens.
And the breeding is highly selected generation after generation.
So it takes over the colony like wildfire. Okay?
Wow.
Now here's the kicker though, where this comes back to your point. Those animals with their broken telomeres, doesn't have to be that way. I can describe to you the protocol that would be used to breed them without causing this problem. To this day, as far as I know, they're still bred with the same protocol. But it breaks them from the point of view of studies of really important stuff, right? Cancer, wound repair, aging, and, most importantly, drug safety. We use these animals to test drugs to see if they're toxic. The problem is, if you have ultra-long telomeres and therefore your tissues can replace themselves indefinitely, if you give them a poison and it doesn't outright kill them, not only does it not show up as toxic, but it may even make them live longer because they are all brewing tiny little tumors, and if you give them a poison, it functions like chemotherapy. Okay? So, when I realized this, mind you, I now realize naive graduate student. When I realized this, I thought, I know they're not going to like it, probably aren't going to like me, but they have to fix it.
No, they don't.
Yeah. Well, not only no, they don't, but they have a reason not to, right? They actually have two reasons not to. When I approached Carol Greider, who has pretended that this whole episode didn't happen and stopped talking to me and didn't cite my work when she published her test of my hypothesis, but when I contacted her and I was like, Carol, this is incredible. Where are you going to publish that work so I can cite it in my paper, my evolutionary paper on this topic? And she said something that now rings in my ears. She says, we're actually going to keep it in-house. I did not know what she was talking about. Why would you have... She ran this experiment that was going to be absolutely a bombshell. The mice we're using for all of our mammal studies are broken. That was going to be a big paper. Why would she keep it secret? Well, the answer is it's actually like having a crystal ball. If you know how the mice are broken and nobody else understands it, then you can predict experiments like you have an insight into the biology, when really you just have an insight into the brokenness of this one apparatus. So there's a perverse incentive not to fix the problem, and by not fixing the problem, our drug safety system, as far as I know, still uses these animals which are biased in the direction of telling us that toxic drugs are safe. Now, whose interest does that serve?
Of course. So it's perfect. It's just, and it's an element of too big to fail. You know, small errors that are slightly advantageous for these mega industries, you know, maybe if there's a bit of spotlight, let it go. But not big ones. Not big ones.
This one actually, the Vioxx scandal.
Oh, yeah.
Right? Here you have a drug thought to be safe, released into humans, does heart damage, which I don't think is heart damage. It does body-wide damage, and we notice it in the heart because the heart is special. But, okay, so you've got a drug, creates a major scandal. How did that get through drug safety testing? Fen-phen, Seldane, erythromycin. We have a long list of drugs that have had that exact pattern. Seemingly safe, released into people, turns out they do heart damage. My claim is that is actually predicted by this model based on broken mice and their ultra-long telomeres. Federal government did a study, released a 300-page book on the future of drug safety. Doesn't mention mice, doesn't mention the genus mus, doesn't mention telomeres. Ignores the entire topic.
Absolutely. And then it just triggered the memory of one of the COVID vaccines, wasn't it? 9 mice were used, and they ran with it based on 9 mice.
May they rest in peace.
Oh my God. Well, here, you know, Bret, this has been superb, but I'm conscious of your time, so